Good day, everyone. Our lesson for today is about translation. In this lesson, you will learn how proteins are made from the code carried by the messenger RNA. In the first part, you will be introduced to the structures and functions of the essential components of protein synthesis, like the messenger RNA, the tRNA, and the aminoacyl-tRNA synthetase, and the ribosome. Within the second part, the molecular details of the overall process of translation will be elaborated. At the end of this lesson, you are expected to identify the molecular machinery that governs protein synthesis, describe the structure and functions of each of these machineries, and discuss the overall process of translation. So before we proceed with our discussion proper, I want you to, we will be having a brief review first of what you have learned during your genetics course. So you have here a messenger RNA that needs to be translated into a protein. So you provide the polypeptide chain encoded by the given mRNA by referring to the genetic code provided. So you use the three-letter symbol for each amino acid. So we know that uh, in translation, now the messenger RNA contains the codons, which are read in groups of three nucleotide bases. Okay, so each of these codons, okay, specifies an amino acid. Okay, so are you already finished translating these messenger RNA? Okay, so you are correct. Now, the polypeptide chain encoded by the given mRNA is methionine, proline, asparagine, glycine, leucine, arginine, and alanine. Okay, so we know this because AUG, A, so AUG, okay, is methionine. Okay, and that is clearly shown here. So A, the first codon is AUG. So the first letter is, the first base is A, second base is U, and the third base is G, so methionine. The second codon is CCG. So the first base is C, second base is C, the third base is G. And that is C, 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 G, and that is proline. So you do that also for the rest of the codons. UAA does not specify a particular amino acid because UAA is a stop codon. Okay, so you have already learned that in your genetics. So the question here is, how is the cell able to use the code that is carried by the messenger RNA to synthesize this polypeptide chain? So we will be discussing into the molecular details of this process. Translation is a process wherein the information contained within the order of nucleotides in messenger RNA or the mRNA is transcribed from the genes interpreted to produce the linear sequence of amino acids in polypeptides or proteins. Okay, so we know that the DNA is transcribed into an RNA and then RNA is translated into a protein. So scientists thought that it would seem unlikely that direct interactions between the messenger RNA template and the amino acids could be responsible for the specific and accurate ordering of amino acids in a polypeptide. Thus, in 1955, Francis H. Crick proposed that before their incorporation into the polypeptides, amino acids must attach to a special adapter molecule that is capable of directly interacting with and recognizing the three nucleotide long coding units of the messenger RNA, because it will be impossible for the protein to immediately attach to each other based on the template or be based on the messenger RNA. So something must act as an adapter to where the amino acids will be attached first. So Crick imagined that the adapter would be an RNA molecule because it would need to recognize the code by Watson-Crick base pairing rules. And it was shown by Paul Zamignek and Malon B. Hogland that amino acids are attached to a class of RNA molecules before they are incorporated into proteins. So these types of RNAs are called the tRNAs or the transfer RNAs. They are named as such because the amino acids attached to them are subsequently transferred to the growing polypeptide chain. Now, there are four primary components that compose the machinery responsible for translating the language of mRNAs into the language of proteins. So what are these components? So these components are the messenger RNAs, the transfer RNAs, the aminoacyl-tRNA synthetases, and the ribosomes. Together, these components translate a code written in a 4-base alphabet into a second code that uses the 20 amino acids to write the language. 4-base alphabet because there is in messenger RNA you have the adenine, the cytosine, the guanine, and the uracil. So the messenger RNA carries the information to be interpreted by the translation machinery and serves as a template for the translation process. The messenger RNA that codes for the protein consists of an ordered series of 3 nucleotide units we call the codons. Atong gikwento before, the AUG, CCG, these are called codons. And they specify the sequence of amino acids. The tRNAs provide a physical link between the amino acids being added to the growing polypeptide chains and the codons in the mRNA. And the enzymes called aminoacyl-tRNA synthetases are the ones that couple amino acids to specific tRNAs that recognize the appropriate codons. Finally, the major player in translation is the ribosome. These are the ones that coordinates the correct recognition of the mRNA by each tRNA and catalyzes peptide bond formation between the growing polypeptide chain and the amino acid that are attached to the selected transfer RNA. Okay, now let us talk about each one of these components in detail. First are the messenger RNA. So the messenger RNA are the one that is produced after transcription. The protein coding region of each mRNA is composed of a continuous and non-overlapping chains of codon called the open reading frame, which specifies a protein. Okay, so when we say non-overlapping, meaning you don't read the bases over and over again, you just read it once. So you have here, for example, this is an overlapping code, AUU, and then UU is read again, UUG, and then UGC is read again. Okay, but in a non-overlapping mode, if this is the codon, so AUU, okay, and GCU, so in an overlapping AUU, and then UUG, and then UGC, and then GCU. So this is an overlapping because the base is read again in the next codons. But it was discovered that the codons is non-overlapping. Okay, meaning you just read it once. So AUU, that is one codon. GCU is another codon. And CAG is another codon for a specific amino acid. Okay, so each ORF, or the open reading frame, starts and ends at internal sites that are embedded within mRNA. This means that the ends of an ORF are distinct from the ends of the messenger RNA. Okay, so you have here a messenger RNA produced. We know that it has a prime cap and a poly A tail, and it has still an ORF with an end with a starting bases and an end. So there are still untranslated regions within a messenger RNA. So translation starts at one end, the 5' end of the ORF, and continues one codon at a time, the end, which is the 3' end. Now eukaryotes, you always use the AUG as their start codon. Bacteria usually use this, but they could also use GUC and sometimes even UUG. So the start codon is one of the functions for specifying the first amino acid to be incorporated into the growing polypeptide chain. And it also defines the reading frames for all subsequent codons. On the other hand, the stop codons UAG, UGA, and UAA define the end of the open reading frame and signal the termination of the polypeptide synthesis. In all organisms, mRNAs contain at least one ORF. Okay? So the ORFs, or that corresponds to a particular protein, okay, is their number is different between prokaryotes and eukaryotes. Eukaryotic mRNAs almost always contain a single ORF. No? So only one open reading frame, only one protein is translated. This is referred to as monocistronic mRNA. But in prokaryotes, now they frequently contain two or more open reading frames, so they could produce two or more protein in one messenger RNA with one promoter region and stuff in the original gene. So these types of mRNAs, now those mRNAs that contain multiple proteins, are known as polycistronic mRNAs. Okay? Again, in eukaryotic mRNA, one ORF, one protein only in one messenger RNA. It's called monocistronic. In prokaryotic mRNA, one mRNA could have multiple open reading frames, so they could produce multiple protein. These are referred to as polycistronic mRNA. So the recruitment of the ribosomes to the mRNA is necessary so that translation can occur. So to facilitate the binding by a ribosome, many prokaryotic ORFs contain a ribosome binding site, or the RBS. So you have here an mRNA with the ribosome binding site, or the RBS. This is a short sequence of the start codon. Is usually referred to as the Shine-Dalgarno sequence. Okay, so the RBS typically located 3 to 9 base pairs on the 5' side of the start codon. Okay, start codon here, and this is the RBS. This is complementary to a sequence located near the 3' end of one of the ribosomal RNA components, the 16S rRNA. So the RBS base pair with this RNA, thereby aligning the ribosome, okay, with the messenger RNA at the beginning of the ORF. The core of this region of the 16S RNA has the sequence CCU, while prokaryotic RBS is most often a subset of the sequence AGG. So you have here, no, the 16S RNA that binds to the ribosome binding site upstream of the start codon, so that they will be perfectly aligned, no, at the catalytic site of the ribosomal subunit. Unlike that of prokaryotic eukaryotic mRNAs recruit ribosomes through a specific post-transcriptional chemical modification called the 5' cap, as what we have talked earlier, which is located at the extreme 5' end of the messenger RNA. So in a previous lesson, we know that the 5' cap is 7-methyl guanosine. Once the ribosome is bound to the messenger RNA, it moves into a 5' to 3' direction until it encounters the start codon. So this process here is called scanning. Okay. So that was all for the messenger RNA. How about the transfer RNA? So the center of protein synthesis is the translation of the nucleotide sequence information into amino acids. This process is done by the transfer RNA molecule that acts as adapters between the amino acids that they specify and the codons. So there are several types of tRNA molecules, and each of these types is attached to a specific amino acid, and each of them recognizes a particular codon or codons in the messenger RNA. All tRNAs have different features in common. Okay, so first, all tRNAs end at the 3' terminus, okay, with the sequence CCA. You have here CCA, which is where the amino acid is attached. The second is the presence of several unusual bases. No? Here in the primary structure, which is created post-transcriptionally by enzymatic modification of the normal bases na in the polynucleotide chain. So in advanced molecular biology, we will talk about, actually, if you enroll in higher molecular biology subject, one of the topics will be the post-transcriptional modification of the transfer RNAs. So example of this post-transcriptional modification of transfer RNAs is the incorporation of the pseudouridine and dihydrouridine, which are derived from uridine bases. Okay. So tRNA molecules show characteristic and highly conserved patterns of single-stranded, double-stranded regions due to self-complementarity, because there are regions that are complementary to each other, so they subsequently fold, actually. Okay, so that's why it is illustrated as a cloverleaf structure. So the principal features of the tRNA cloverleaf are the acceptor stem. Okay, the three stem loops referred to as the U, the D, and the anticodon loop, and the one which is the variable loop. So again, acceptor stem, the stem loops, the D loop, the U loop, and the anticodon loop, and the variable loop here. So what are the functions of these different parts of the tRNA? The acceptor stem, okay, is the attachment site for the amino acid, no, because it has the sequence CCA. It is formed by pairing the 5' and the 3' ends of the tRNA molecule. The 5-CCA protrudes, no? This protrudes. So we call this an overhanging end. And is preceded by unpaired discriminator base. Okay, the U loop is the most 3' proximal position. Okay, and contains pseudouridine residues frequently found in the sequence, no? TUGCG. D loop, the most proximal loop, contains dihydrouridine residues. The anticodon loop here contains the anticodon sequence that is complementary to the mRNA codon. Okay, so they actually interact by RNA-RNA base pairing. So the variable loop here is located between the anticodon loop and the U loop. It varies size from 3 to 21 nucleotides. Now, cells do not have a specific tRNA for every codon, no, as this would require 61 tRNAs because for every codon we know that they specify a particular amino acid, and there are 61 combinations of the 61 possible combination in a 4 alphabet na bases of the DNA. So it will be impossible, no, to have one tRNA for every codon. So instead, it was discovered that certain tRNA species of known sequence can recognize several different codons. Thus, the genetic code is described as degenerate. So the degeneracy of the genetic code means that one amino acid could be specified, no, by, or it could be coded by several codons. So if we go back to the genetic code that I've shown you earlier during the start of before the start of the discussion, you will notice that several codons could code for the same amino acid. So that's why we call the degeneracy, no, of the genetic code. Furthermore, some tRNAs do not have one of the 4 classical bases but have inosine instead in the first position of the anticodon. Like all the minor tRNAs, inosine arises, no, through enzymatic modification of bases in a completed tRNA chain. So, the wobble theory developed by Francis Crick in 1966 states that the base, not the 5' end of the anticodon, is not spatially confined, no, as the other two, allowing it to form hydrogen bonds with any several bases located at the 3' end of the codon. So this is because sliding can occur at that alternative hydrogen bond formation is possible. However, not all combinations are possible. The pairings that are allowed by the Wobble theory are those that produce ribose-ribose distance close to that of the standard EU and GC base pair. So, pwede lang. As long as they are at the position where the anticodon loop curves around. So, pwede na siya. But the streak there should be the center basis of the anticodon loop. The third component are the aminoacyl-tRNA synthetases. Okay. So, charged tRNA molecule. So, when do you say that a tRNA molecule is already charged? If an amino acid is already attached to a tRNA, that means the tRNA is already charged. So, charged tRNA molecules are those tRNAs with an already attached amino acid. The requirement for charging are the acyl linkage between the carboxyl group of the amino acid and the 2' or the 3' hydroxyl group of the adenosine nucleotide that protrudes, okay, from the acceptor stem at the 3' end of the tRNA. So the enzyme responsible for charging the tRNA or for adding this amino acid into the tRNA are called the aminoacyl-tRNA synthetase. So they're responsible for that catalysis. So two enzymatic steps are performed by all aminoacyl-tRNA synthetase during the attachment of an amino acid to tRNA. So step 1, the adenylation in which the amino acid reacts with ATP here to become adenylated with the concomitant release of a pyrophosphate here. Okay. So adenylation refers to the transfer of AMP as opposed to adenylation, which is the transfer of adenine. Okay. Step 2 here, okay, you can read through the process of the charging of the tRNAs, okay, as what is detailed here. Now the question is, how does the aminoacyl-tRNA synthetase recognize the correct amino acid to which they would charge into a particular RNA? Well, how do they know that this is the right amino acid? We know that specific amino acids carries a specific anticodon that will recognize specific codon. So how do they know that, for example, the codon is AUG? So the anticodon on the tRNA should be what? UAC. So how do they know that the methionine is really attached to a tRNA that contains the UAC anticodon? So studies have indicated that determinants for the specificity of tRNA recognition by aminoacyl-tRNA synthetase are found at two distinct sites in the molecule. Now, the acceptor stem and the anticodon loop. So the aminoacyl-tRNA synthetase, the enzyme, must couple the proper amino acid with its cognate tRNA molecule because the ribosome cannot distinguish between correctly and incorrectly charged tRNA. So it is at the aminoacyl-tRNA synthetase function that the correct amino acid must be attached to the correct anticodon because there is no way the ribosome could discriminate which one has the proper amino acid attached. Okay? So we have already discussed about the messenger RNA, the transfer RNA, and the aminoacyl-tRNA synthetase. Now let us talk about the ribosome. So directing the synthesis of protein is the macromolecular machine called the ribosome. So it is a larger and more complex than the minimal machinery required for DNA and RNA synthesis. In prokaryotes, the ribosomes can commence or they can start immediately the translation even before the messenger RNA is already finished being transcribed by the RNA polymerase during transcription because they are just located in the same compartment. There is no separation of the process in prokaryotes because they have no organelles. In eukaryotes, on the other hand, the transcription occurs in the nucleus and the translation occurs in the cytoplasm. That's why the transcription must be finished first. The messenger RNA will be carried to the cytoplasm for translation to occur. Unlike in prokaryotes that even though transcription is not yet finished, they can already start translation. So the ribosome is composed of an assembly of RNA and protein known as the large and small subunits. The large subunit contains the peptidyl transferase synthetase. So this is the catalytic site where the formation of peptide bonds between the amino acids are occurring. The small subunit, okay, contains the decoding center in which charged tRNAs read or decode the codon units of the mRNA. By convention, the large and small subunits are named according to the velocity of their sedimentation when they are centrifuged. Okay, so at which velocity does a particular tRNA settles at the bottom. So that is the unit S. Now the S refers to the Svedberg. Okay, so you have 16S, so that does not mean subunit, it means 16 Svedberg. Okay, that's the unit of velocity of their sedimentation. Okay, so you have here a comparison of the prokaryote and eukaryote. So you have here the small subunit in prokaryote 16S RNA, and you have here the tRNA plus proteins. Okay, so 16S plus proteins are total of 21S. Okay, 23S RNA and proteins 31 RNA. So, however, if you combine them, it will only result to 70S. So 30S small subunit and 50S large subunit in prokaryotic, but the combination is 70S because the Svedberg is not additive. Okay, the same thing with eukaryotic. 40S small subunit and 60S large subunit, a total of 80S. Okay, they are not again additive. That depends on how they will sediment during centrifugation. Now, how is the peptide bond formed in the growing polypeptide chain? So the ribosome also catalyzes the formation of a peptide bond, which is called the peptidyl transferase reaction. So this enzyme reaction occurs between the amino acid residue at the carboxy terminal of the growing polypeptide and the incoming amino acid to be added to the chain. The growing chain attached to tRNA is called the peptidyl tRNA. Okay? While the tRNA with attached amino acid is called aminoacyl-tRNA. So again, the tRNA attached to the growing chain is called the peptidyl tRNA. The tRNA with an attached amino acid is called aminoacyl-tRNA. The ribosome must be able to read at least two tRNAs simultaneously to perform the peptidyl transferase reaction. Okay, so the ribosome contains three binding sites called the A, the P, and the E site. Now it is at the A site responsible for the binding of the aminoacyl-tRNA. The P site is the binding site for the peptidyl tRNA, and the A site is the site for binding the tRNA that is released after the growing polypeptide chain has been transferred to the aminoacyl-tRNA. So what is the overall process of the translation? So you have here the process of translation in prokaryotes. I know this is a prokaryote because you have here an FMET. FMET is for methionine in bacteria. Okay. So you have here the E, the P, and the A sites. You have the messenger RNA, and you have the ribosome, the small and the large subunits. So again, these are recruited via the shell, the Garnos sequences that is attached to the particular site here in the ribosome, so that they will know how to align themselves. The tRNA that contains the methionine, amino acid methionine, recognizes the AUG, and then the addition of another, they will advance to the A site. Okay, the A site, and then at the P site, another no, the an aminoacyl-tRNA binds to the A site, is courted by the AF2 bound to GFT protein. During the tRNA binding, the GTP is hydrolyzed and released here. Okay. At the A site, the another tRNA that contains no the that contains another amino acid no that is corresponded by this codon no will attach at the A site. The peptide bond formation no will occur via the peptidyl transferase reaction, and then the the amino acid will be released from the first tRNA, and the tRNA they will move the ribosome will move one step ahead, one codon ahead, and will recognize another codon. The tRNA that carries the previously the first amino acid will be released. Okay, so uh another transfer RNA containing amino acid that's corresponded by this codon will bind to this, and so the whole cycle is just repeated. Okay, overall the process it needs the action of the enzyme and is driven by the several energy expenditure. Okay, so in this lesson we have just discussed about translation. What are the different components and the molecular details of the molecular structure of these different components, as well as the overall mechanism of the translation process. So again, to summarize this, it will be helpful in your review, no? So you need to answer these questions. So what is translation? What are the important translation machineries? What are the molecular mechanisms of their function? What are the stages of translation? And discuss the molecular process. So if you have further questions or any questions to clarify the lesson, you can contact me or your lecturer through their provided email addresses. You can also directly key in your question at our or your group chats. Okay, thank you very much and see you again in our next lesson.